You should be seeing my screen now. Do I need this? No. Let me briefly talk about the unknown project. If you have a request for a test, remember to write it up in the bottom of your unknown notes page that you submit. I am not going to take requests over the email. And if you do email me, I'll politely tell you to put it in your unknown notes page. Today I went through the unknown notes page, and every test that needed to be sent out has been sent out. There is one or two test requests where the tests are incubating, and the results will be sent out after the test has incubated. And you can look under week 5, let me show that, in the lab, week 5 lab, scroll down, and it has the different tests that you can look up how long the incubation has to be for. Any questions about any of that? All right, so put everything in your unknown notes page. Most of you have at least your unknown number in the page. You should also have that you ran the gelatin hydrolysis test last Tuesday, not this Tuesday, but a week ago on Tuesday, and the gram stain last Sunday, and the lactose fermentation test on Monday. And on Tuesday night, late Tuesday night, I did send out the results. About half of you have your results up. So please put your results up on those 3 tests in your unknown notes. And then take a look at the chart. It's right here. Unknown list. I never remember the name of that. Unknown list. Spring 2020, meaning I haven't changed it since then. And it has the chart of all the possible species we're going to look at and all the tests we're going to look at. You don't have to run every test, obviously. You only need to run the test you need to identify your unknown. Any questions about any of that? Okay, no questions. I was afraid I was going to be behind schedule, but it turned out I got everything out before class today. Let's take a look at the syllabus. What we're doing today is we're talking about microbial growth, chapter 6. We're supposed to finish this lesson today. So I'm starting it and finishing it. And it will be on the quiz next Wednesday, which will cover chapters 5 and 6 and lab 7. Any questions about any of that? There is no lab today, so you're going to be working on your unknown notes. I will show up at 6:30 for 15 minutes to take questions. But at 6:45, if there are no further questions, I will log off. You can email me questions if you have a question after that time. All right, any questions about any of this? Let's jump into the lesson. Like always, I show you, I'm not hearing anything from students. Is everything clear to everyone?
Yes.
Thank you. I always start out with a slide showing you the major goals for the lesson and the rough outline. As always, know the terms. I'm not going to speak about them this time because I'm trying to go through this lesson rapidly. Understand the various requirements of bacterial cells for growth, and we are going to talk about a few other cells, and you can use these requirements for organisms, but we're really going to only look at it in this lesson for bacteria. Understand the 4 phases of growth, and then compare the different methods for measuring microbial growth. When we're talking about microbial growth, you should understand that we mean an increase in the number of cells, not an increase in the cell size. Any question about that? Come on.
No.
Let's take a little look at the time lapse. That'll take a while, so let me discuss it. Oops. There are various requirements for cells to grow, including physical and chemical requirements. And we'll get back to that topic.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right, any questions about that? Showing you.
Yeah, what does it mean when she says billion?
That means a billion billions. So 10 to the 6 times 10 to the 6. Personally, that seems a bit high for me in one day, but I know they can get up to, they said a billion, didn't they?
Yeah, that's what I was like. That sounds
That
like
doesn't
extreme.
make sense. Yeah, that's too high. It can get up to maybe 10 to the 9th cells in a day. And I think a billion would be 10 to the 12th. But that sounds like it's too high. I think it can get up to 10 to the 9th cells in a day. Anyways, I'm not going to quiz you on that. The point is that bacteria simply split into 2, and if they're reproducing every 20 minutes, they can produce a large number of cells in a single day. For our purposes, for the fast-growing cells, one cell or maybe a clump of cells on a petri dish in a day can grow up into a pretty large colony. And by large colony, I know we're not going to go over that today, but a large colony, let me show it to you. A large colony is one where it has, there it is, the size of the eraser of a pencil or larger. Anyways, the various requirements for microbial growth. Physical requirements include temperature, pH, salinity, osmotic pressure coming into play with salinity. Various chemical requirements. All living things, including bacteria cells, require a carbon source, a nitrogen source, a source of sulfur, phosphorus, trace elements, oxygen, and organic growth factors if they require them. Now, not all living things require these. For example, if we had a photoautotroph growing in sunlight, what would they require? A photoautotroph. Okay, nobody's answering, so are they going to require a carbon source?
CO2?
That would be their carbon source, meaning they don't require any because they can get it from the carbon dioxide in the air or the water. Would they require an energy source? That's not listed
Isn't it light?
here. Yeah, they're getting it from light, so they don't require any other energy source. If they fix nitrogen from the air, they don't need a nitrogen source, but they would need a sulfur source and a phosphorus source and trace elements. Trace elements, things including us, bacteria cells, and us, generally get trace elements from the water they're around or consume, or for us, we get trace elements also from the food we eat. That would be things like iron, copper, magnesium, and we generally get enough of those in the food we eat and the water we drink and anything else we drink. And then bacteria get it from their environment as well. Okay? Oxygen, of course, we breathe that in, and bacteria get it if they need it through diffusion. Organic growth factors, bacteria could require those. An organic growth factor that we require would be vitamin C, as well as almost all the other vitamins. Various physical requirements. Let's start off with temperature. Each species of bacteria has its own minimum temperature, maximum temperature, and optimum temperature that it will grow at. The optimum temperature is where the species will grow the fastest. The minimum temperature it'll grow at, and the maximum temperature it'll grow at. Above that temperature, the bacteria would not grow. Any questions there? We can classify. Thank you. We can classify microorganisms into their temperature groups, the preferred temperature that they like to grow in. And with those, we've got the psychrophiles. These are the organisms that grow at 10 degrees Celsius or a little less, but not usually down to freezing. They'll have to have an antifreeze in them to grow below 0. And there are some species that do make antifreeze, and generally they can grow at temperatures down to about a minus 10 degrees Celsius. These would be the organisms you find in your refrigerator, and they can grow and cause your food to spoil. They would take usually about a week to grow and cause the food to spoil. Why, once the food is a week old in your refrigerator, you should throw it away. And that's not to say foods that are preserved with salinity, like salted fish, or both vinegar or pH, low pH, and salt, like ketchup or pickles. They can stay in your refrigerator much longer than a week. And don't ask me what the greatest length of time they can be. I think we had a bottle of ketchup that was over a year old. It might have been about 2 years old. And I told my mother we should give that to my brother Mike because he really likes ketchup and we don't use it. And so we gave it to him and he used it up, and he's still alive, so it didn't kill him. The next organism are psychotrophs, and these organisms have an optimal temperature around 20 degrees Celsius, but they can go down, maybe down to about 0 degrees. And I'm not sure what these are. Things that grow at room temperature or less. Mesophiles are the organisms that grow best around 37 degrees, and that's our core body temperature. These are the organisms that are human pathogens. These organisms can make us sick, get in our body, and infect us. The psychrophiles, if they were to get into your body, like when you eat spoiled food, and you get the psychrophiles from eating the food in the refrigerator, they're not going to grow in you. In fact, their membranes and their enzymes will fall apart because they're designed to grow at 10 degrees Celsius or less. And at 37 degrees Celsius, it's much too warm for the psychrophiles to grow and cause an infection in you. In fact, that's true for all of these trophs. The only one that will grow in you are the mesophiles. The thermophiles and the hypothermophiles, if they get in you, their membranes are literally going to freeze up, their enzymes are going to contract too much, and they're not going to grow in you. Only the mesophiles can grow in you and cause infections. The mesophiles, like I said, grow at 37 degrees. Let me state that these definitions are not absolute, meaning every species varies. Micrococcus luteus grows on our skin. Its optimum temperature is not 37. Its optimum temperature is 30 degrees because our skin is a cooler temperature than our core body temperature. Any questions about any of that? The point is that these aren't rigidly defined. The mesophiles are anything that grows near 37 as their optimum, and the psychrophiles anything that grows near 10 degrees. And then the range especially can vary for each species.
Are there any microorganisms that can grow at any temperature from like -10 to all the way up to 110 degrees Celsius?
No, there's no microorganism that has that big of a characteristic. There are some that have a larger characteristic, but it's never as large as 2. Like there isn't something that grows from 40 degrees Celsius to 110 degrees Celsius. Okay, but there are some, I don't know, that might grow from 60 degrees Celsius to 110 degrees Celsius. And that's quite a big range.
Thank you.
Okay. All right, the thermophiles are things that grow at 60 degrees Celsius or maybe 62 degrees Celsius. And if you ever look at the chicken at Safeway going around and around on the rotisserie under the heat lamp, those organisms growing in that will be the thermophiles. And the thermophiles, when they've grown for a period of time, it's much faster than a week because it's not cold, so the organisms can grow rapidly. But anyways, the point is, if you see the chicken starts to look old or drying out, you probably shouldn't buy it because it's been under that heat lamp for too long. And the thermophiles can grow and cause food spoilage. The hypothermophiles are organisms that grow at 95 degrees Celsius or around that temperature. Remember, 100 degrees Celsius is the temperature of boiling water. And surprisingly, we don't know any hypothermophiles that cause illness in people. And we know that people are exposed to hypothermophiles, not often, and most people are not, but there's some people in villages in Mexico, and they have a, what do you call that, a stream that's a magma stream. What am I trying to say? Not a deep sea vent, but like a glacial, no. Like a small Yellowstone with the magma hot streams. Do you know what I'm talking about?
There?
Anyways, they take their corn and stick it in a gunny sack and then throw it in the boiling water or close to it where the magma is heating the water, the hot streams, and cook their corn and then pull it out. And there's hypothermophiles all over the corn because they're all over the water there. And then they take it home to their family and eat it, and it does not make the people sick. Okay? Anyways, the hypothermophiles cannot grow in us, and there's none known that actually cause people to get sick. I'll just word it that way. All right, any question about any of this? Showing you the different trophs for what temperature an organism optimally grows at. Okay? We talked a little bit about food spoilage earlier. Let's talk a little bit about it more. Let me see if I can blow this up a little. Anything in the red zone is the rapid growth of bacteria. And so once the food is sat out at 60 degrees Fahrenheit to 130 degrees Fahrenheit for more than 4 hours, you should not eat it. After 4 hours, that food, well, should be tossed. So you don't want to have your, I don't know, your meat or things sitting around at room temperature, which would be 68 to 72 degrees Fahrenheit, for more than 4 hours. Put it in the refrigerator once it cools down, and that will keep it safe because that will be down here. And organisms can grow out of a temperature beyond 60 to 130 degrees, and they actually grow down to about 38 degrees, but very slowly in this range. And that's why in the refrigerator, the meat and other things, after it's been in the refrigerator for a week, you should throw it away. Any question about that? Food spoilage can occur by mesophiles growing in the food before the temperature of the food gets colder, the refrigerator, and by psychotrophs, the organisms that grow in between freezing, they're the very cold organisms, and the mesophiles. Food spoilage occurs when it's held at too warm a temperature or when it is in too large of a vat and it's added to the refrigerator, but because it's such a large amount, the food doesn't cool down quickly enough, and then organisms can grow in it. And usually the organisms that grow in it release a toxin. And so the food looks fine, but it has a toxin in it. And then when people eat it, they get gastroenteritis. All right, this is the start of the in-class activity, which is the discussion worksheet, talking about the physical requirements that bacteria grow. And this one's talking about the pH. And I'm going to skip over that because you're doing that in the worksheet. Various physical requirements, chemical requirements. Oxygen, which is actually a toxic substance, even for aerobes. There are certain oxygen compounds or metabolites which are toxic. And if you're growing in oxygen, then you have to be able to detoxify these toxic oxygen compounds, or else those toxic oxygen compounds will make you sick and then kill you. And that applies to both people as well as microorganisms. All right, so this is after the worksheet. A various chemical requirement that we did briefly discuss. Let's go on to it further. Oxygen. Microbes that use oxygen, they're called aerobes, require oxygen in order to make ATP. And they produce more ATP than other organisms from their nutrients that don't use oxygen. So aerobes tend to grow faster than anaerobes. Microorganisms are classified based on the effect oxygen has on their growth. How we test the effect that oxygen has on the growth of a microorganism is we make a thioglycolate tube. Thioglycolate is a media that removes oxygen from the media. And when we autoclave the thioglycolate tube, when it immediately comes out of the autoclave, there's no oxygen in the media. But oxygen from the air moves into the media so that the thioglycolate tube will have an oxygen concentration. At the top of the tube, there will be full oxygen, about 20%, 20 or 21, I don't remember, but full oxygen from the air. And as you go down the tube, there will be less and less oxygen till you reach the bottom of the tube, where there will be no, or essentially no, oxygen in the tube. Any questions about that? So organisms that only grow at the bottom of the tube are anaerobes, and if they're only growing at the bottom, we call them an obligate anaerobe, meaning an organism that cannot grow in the presence of oxygen. And that's not shown very well here. The majority of the growth should be at the bottom, and as you go up the tube, there will be less and less growth. So there should be a 3 there, and then a 2, and then a 1, all near the bottom of the tube. Any questions about that? All right. So the next easiest is the aerobes. Obligate anaerobes can only grow where there's oxygen, and they will have their growth at the top of the tube where there is oxygen. The most growth will be at the very top where there's the most oxygen. And as you go down the tube, there will be less and less growth until you get to the point where there's very little growth and very little oxygen. And below that, there's no growth. Any question there? Excuse me. It's snowing lightly here, and for some reason it's giving me a bit of a allergy. The next one we're going to talk about are the facultative anaerobes. They can grow with oxygen, and then we'll use oxygen in aerobic respiration, but they don't require oxygen, and if oxygen isn't around, they fall back on some other method of making ATP that doesn't require oxygen, and that could be anaerobic respiration or fermentation. In these facultative anaerobes, you'll see the most growth at the very top of the tube, because these organisms are growing by aerobic respiration, and they're going to be able to make more ATP than the organisms growing at the bottom of the tube, which have no oxygen. So they're growing by either anaerobic respiration or fermentation. And as you go down the tube, you'll see less and less growth. So we'd see 3 right here, 2 right here, and then 1 at the very bottom. Any question about any of that? All right. There's also aerotolerant anaerobes. These are anaerobes, so they do not require oxygen for their growth. So you will see the growth of an aerotolerant anaerobe everywhere in the tube, and they will be distributed evenly throughout the tube, meaning, I don't know, 3 here, 3 there, 3 there, 3 at the very bottom. How they're able to grow with oxygen, since these are anaerobes, is they have all the enzymes they need to detoxify the toxic oxygen molecules, the toxic oxygen radicals. And you can learn about that in the worksheet. there would be enzymes such as SOD, superoxide dismutase, catalase, peroxidase, enzymes like that to detoxify the toxic oxygen metabolites. And as you can imagine, aerobes have to do that as well. And we, as well as bacteria that are aerobes, do that. We have catalase, we have SOD. I don't think we have peroxidase, but some bacteria do. I don't think I ever asked the question, why do the facultative anaerobes grow best at the very top of the tube, and then their growth slows as you go down the tube? So I'm asking that. Why do they grow best at the top of the tube?
More oxygen on top.
Yeah, there's more oxygen, so they can perform aerobic respiration, and they're going to grow fastest when they're performing aerobic respiration, because that makes the most ATP. The ones making less ATP are going to be further down in the tube where there's less oxygen, or maybe the bottom where there's no oxygen. All right, the last one we need to talk about are the microaerophiles. These are aerobes, so they do require oxygen to make ATP, but they do not require, or they do not prefer, full oxygen. And they usually grow best between 2 and 10% oxygen, which is less than full oxygen from the air. And these will grow someplace in the middle of the tube, at least best at the middle of the tube. Any question about that? A microaerophile is Helicobacter pylori, which is a bacteria that grows in your stomach, and it can cause ulcers. Your stomach, from the bacteria in it, use up the oxygen, and they reduce the oxygen in your stomach, and Helicobacter pylori is one of them. Any question about that? All right, let's move on. When we talk about culture media, media is a plural term for several different media. When we're talking about one, how do you say that? One of the media, then it is said a medium. So medium is singular, media is plural. It's a liquid or a solid solution that provides all of the requirements for an organism to grow. You know some general purpose media like TSA or NA, nutrient agar with yeast extract. And they're liquid if they are in a broth, meaning they're liquid. And to solidify that, we use a solidifying agent, usually agar, but there could be other solidifying agents such as starch or gelatin. When we give it to you in the lab, it's sterile. That means the medium has no living organisms in it, including microbes, and it's usually sterilized in an autoclave. And then you guys inoculate that medium. The inoculum is the microbes introduced into the medium. The culture is the microbes growing in or on that culture medium. So we call things growing in the culture media a culture. That's a little bit confusing. There's a very wide variety of media that are available to you. And if you were in the lab, I would hold up a book of various media. I don't know if you can see that. There we go. It's about that thick. And each page of that book discusses one medium. So that's an awful large number of media that are available in microbiology. Any questions about that? We'll never talk about that many, but we do talk about a few media. So the purpose of media is to support microbial growth. By doing that, the medium must provide an energy source, a carbon source, sources of nitrogen, sulfur, phosphorus, trace elements, and organic growth factors. Oftentimes, the energy source and the carbon source can be the same molecule, like if you're providing, what is the name of that? Glucose salts media. Glucose is both the energy source and the carbon source, and I think it might be the nitrogen. No, there's no nitrogen in glucose. Sorry, I'm getting confused. There has to be a nitrogen source and a sulfur source and a phosphorus source in addition. The trace elements frequently are not added to the media, that we simply add the trace elements from the water. And some trace elements come from the powder of the media. Maybe not glucose salts media, but if we were to add beef extract to the media, the beef extracts, meaning you're getting the extracts from the cow, meaning the trace elements from the cow. Is that clear to everyone? Trace elements frequently are just in the environment, and they may just come from the water. Organic growth factors, we talked about these before. Those are the things that are essential for an organism to grow. For us, it would be vitamin C and almost all the vitamins, the essential amino acids, and the essential fatty acids would be the organic growth factors. Now, to support microbial growth, these things must be added if the microorganism requires them. But as I said, a photoautotroph doesn't need an energy source or a carbon source, but it would need a nitrogen source, a sulfur source, and a phosphorus source, and of course the trace elements from the media or from the water. For most bacteria, we don't need to add an organic growth factor, but sometimes we do, like Streptococcus pyogenes will not grow on a general purpose media like tryptic soy agar or Nay, but it will grow on a blood agar plate, and the Streptococcus pyogenes is getting its organic growth factors from the red blood cells in the blood agar. All right, any questions about any of that? Let me remind you, Streptococcus pyogenes causes strep throat and a whole bunch of other diseases which are not very common anymore because of the development of antibiotics. I think that was it red fever or something like that, scarlet fever, something like that, was something that Streptococcus pyogenes used to cause a lot in the old days, but nowadays those are very rare diseases, although strep throat is still pretty common. Agar is something that we add often to a media to make it a solid media. So agar is used as a solidifying agent for various culture media. And there are a few others, but we're not going to talk about them. Agar is a complex polysaccharide derived from seaweed. You usually find agar in Petri plates or in tubes. There are two types of tubes. Let me illustrate. I really should open paint, but I don't know how to get a button to open paint, so I'm going to use this.
You're muted.
Hopefully I was only muted there for the very end. Oh, come on. Where's my paintbrush? That'll help. Ah. I don't know if that'll work. Now let's draw a shape. Alright, so we have... I'm not sure what's going on here. That's not what I want. Where's the paintbrush, people? I'm having a hard time finding it.
Under design.
Under what?
Design.
Okay, which button do I hit?
At least it used to be.
Let me see if I can get paint open because I'm not finding that here. Oh, come on. Ah. All right. So we have a tube. This is being very long. And if the media is put in like that and it's a solid media, we call that a slant. If on the other hand the media is put in as a liquid and put across straight, that's supposed to be straight across, that we call a deep. And you can have a combination where you have, I'm just going to do it like this, a deep and then a slant, and so the media goes like that and then like that. So you can't have both, but that's a slant and that's a deep. Any question there?
No.
All right, so that's showing you a slant and a deep. Agar is generally not metabolized by microbes, so the media will not be, the agar will not be liquefied, meaning there's almost nothing that can digest agar. Any questions about that? Why agar is often used as the solidifying agent, and that is there's almost no microbes that digest it. When you have gelatin or starch, there are lots of microorganisms that can digest those two as the solidifying agent. And then your media is no longer solid. There are different types of culture media. A chemically defined media is one where we know the exact chemical composition of all of the molecules in the media. Let me blow this up. For example, here is a chemically defined media for growing typical bacteria like E. coli, glucose, supplying both the energy and the carbon source, ammonium phosphate, supplying the nitrogen as well as the phosphate, sodium chloride, providing salinity as well as sodium and chloride, magnesium sulfite, providing the sulfur as well as magnesium, potassium phosphate, providing the phosphate and potassium, and then water supplying the trace elements. And so we know all of the molecules in here, and if you use distilled water, you know exactly all of the molecules that are in this media, meaning there are no trace elements or essentially no trace elements added if you're using distilled water. Any questions there? That's a chemically defined media where we know all of the molecules in there. Many growth factors may be required for organisms that need growth factors, and if they require growth factors, we call them fastidious. So a fastidious organism is not going to grow in this media. This is a media, I don't know if you can see that. Let's see if I can blow that up. Well, let's end the screen. Oh, I see what I did. No, I didn't. No, I don't. Let me end the show. I don't have that now. Ah, here it is. Maybe I did go to the next slide. Try and blow this up so you can see it. An organism that's fastidious requires a lot of growth factors, such as this media on the bottom here. And there's a whole bunch of, things that are added to this media. Streptococcus pyogenes would not grow even with all of these things added, amino acids, organic growth factors, because even that is not enough added to get Streptococcus pyogenes to grow. Like I said, Streptococcus pyogenes needs blood to grow, and we're not entirely sure what all the things are in blood that are being supplied to Streptococcus pyogenes. Complex media is another type of media. It contains extracts and digests of yeasts, meats, or plants, and this provides vitamins and organic growth factors. The exact chemical composition is not known in a complex media. Like a very simple one is this shown here. This is nutrient agar. It has peptone, a partially digested protein, beef extract to grind up the cow and add 3 grams to 1 liter, sodium chloride, agar, and that's all we're adding. But the beef extract, as well as potentially the peptone, will have many different molecules contaminating it. The beef extract will have whatever the cow was eating, and that would be different for a cow in Washington as to a cow in Alabama, because obviously there's different plants in Washington and Alabama. Any question about complex media? We do not know what all the molecules are, because when we add beef extract, you're adding a ton of molecules that were in the cow. The beef extract and the peptone are the primary energy source, as well as supplying the carbon, the nitrogen, and the sulfur. Meaning protein, especially methionine and cysteine, have sulfur in it, and then almost all the amino acids have nitrogen. They all have carbon in it. Oops. I should reword that and say they all have both carbon and nitrogen in it. We use complex media for growing heterotrophs, bacteria and fungi that require an organic carbon source. For the chemically defined media, they're usually not used for anything except for an autotroph, because autotrophs make all of the molecules they need from water and carbon dioxide. Any question about any of that? There are different types of media. We have an anaerobic media, and that's a media that we use for growing anaerobes in. These organisms must avoid the exposure to oxygen. And an anaerobic media is also called the reducing media because it has a reducing chemical in it, like sodium thioglycolate, that combines and removes oxygen from the media. So it has a reducing chemical in it that removes the oxygen. And that results in oxygen depletion. Usually you have to heat the media to drive off the absorbed oxygen in the water in the media. And then you heat it, and then the media is able to remove the oxygen from the media. With the thioglycolate, as I stated, the bottom of the tube, a thioglycolate tube, won't have any oxygen, but as you go up the tube, you'll get more and more oxygen to the very top of the tube where you have full oxygen. There's different types of media. Another different type of media we need to talk about is a selective media. Selective media halts or inhibits the growth of a particular group of microorganisms while not affecting the growth of other microbes. An example of a selective media is 7% sodium chloride media, and this allows salt-tolerant organisms to grow, but it inhibits organisms that don't grow in 7% sodium chloride media, meaning they don't grow in salt. Like Staphylococcus species can grow in 7% sodium chloride media. Streptococcus species cannot grow in 7% sodium chloride media. Most organisms do not grow at between more than 1 to 2% sodium chloride. And so if it grows in 7% sodium chloride media or something higher in sodium chloride, we call that a halophile. Halo means salt, and phile, salt lover. Any question about that? Streptococcus, Staphylococcus, like Staphylococcus aureus or Staphylococcus epidermidis, are halophiles. And you can find them growing on your skin. Although hopefully you won't find Staphylococcus aureus on your skin. But some people have it. There are other different types of culture media. There's a differential media. This media provides differences between different bacterial species or groups of organisms, and the difference is not related to how well the organism grows on the media. Blood agar plate is a differential media in that you can differentiate between the hemolytic activity of different bacterial species. So this species is gamma hemolytic, meaning the red blood cells are not lysed from the media all up until where the cells are. This species of bacteria is alpha hemolytic. The red blood cells are not lysed here, but as we come, we see a dark ring around the growth of the bacteria in the alpha, as well as inside the alpha. It's darker than outside. So this is alpha hemolytic activity, partial hemolysis of the red blood cells. And then this is beta hemolytic bacteria. The B in the beta, the white, is where the bacteria are. And then this clear region is where all of the red blood cells have been lysed in beta hemolysis. So differential media makes it easy to distinguish between colonies of different microbes. Let me do state that blood agar is more than just differential. It's also an enriched media. So a general purpose media that's enriched with red blood cells so that we can grow fastidious organisms on it. like Streptococcus pyogenes. Any question about any of that? Now please understand that a media can be more than one type. I mentioned that blood agar is both differential as well as an enriched media. I think our lab called it an enriched general purpose media. There's also media that are both selective and differential. EMB is an example of that. EMB, eosine methylene blue, is selective against gram-positive bacteria cells. The eosine and the methylene dyes get in the cell and keep the gram-positive bacteria from growing, but they do not select against the gram-negative. So if it grows on an EMB plate, it's likely to be gram-negative. It's also differential on the basis of lactose fermentation. If the organism ferments the sugar lactose, it'll lower the pH, and that will make the cells look darkish or greenish metallish. See the dark blackness on the cells? And that's from the low pH from the lactose being fermented. Other organisms that can grow on the bacteria, on the EMB plate, but do not ferment the sugar lactose, they will be white or off-white in appearance. And there you can see white streaks there too. We already mentioned a little bit about enrichment media. Enrichment media encourages the growth of certain organisms, but not other organisms. So therefore, all enrichment media is selective. It increases the numbers of desired microbes to detectable levels. But an enrichment media is not used to be selective. It's used to enrich for a particular species of interest. It's particularly useful if a targeted microorganism is only a small proportion of the total population. And an enrichment media is often used for isolating soil microorganisms, other environmental microorganisms, or when looking for a rare human pathogen in a fecal sample. You would first put it in the enrichment media, enrich for that species, and then streak out the plate to see if you can get the microorganism of interest. An example of using an enrichment media, let's say that we are wanting to get from the soil a phenol-degrading bacteria. And there probably are a few phenol-degrading bacteria in our soil sample, but there are thousands of other bacteria. If we were to streak it out on a plate, our chance of getting a colony of the phenol-degrading bacteria would be really slim. But if we were to grow it in an enrichment media, such as in a media where phenol is the only carbon and energy source, then the phenol-degrading organisms could grow in this enrichment media, and other organisms that cannot utilize phenol would not grow. So this would be enriching for the phenol-degrading bacteria that came from the soil. And if we can enrich enough, we could then take a sample out of that enrichment media and streak it out on a plate, and we would have a good chance of finding a phenol-degrading bacteria. It might be you'd need to take 1 ml from the enrichment media and put it into a new flask of enrichment media, and might even transfer it each time. Let's see, I still have paint out here. So let's see if I can erase this. And we'll just make 3 tubes like this. You could grow it in the first one and it might enrich for our bacteria of interest by 10 times. And if your bacteria is, I don't know, 1 in 1,000, you only have now 1 in 100, so you'd have to streak out 100 colonies. But if you were to take a mill and put it into a new one, enrichment media, and then we streak it out again, if we were to streak out 100 colonies, we'd have a chance of finding it. But we could enrich again. That might be a little too high if I say 10 times, so let's say 2 times. So that's 100, let's say 5 times. 500 times. And it was 1 in 1,000. Now we only need to streak out 2 colonies, and we'd have a good chance of getting a bacteria of interest. Meaning about 50% of the cells, when we streak it out here, will be cells of interest to us. That's really, really good. Generally, you want at least about 10% of the cells to be the cells of interest. And then you only have to streak out 10 colonies, and you're likely to get 1 colony of interest. Any question about any of that? So that's what enrichment media is used for, enriching for organisms of interest. And you can re-inoculate them. All right, this slide is just telling you the different types of media we discussed. The chemically defined media, where we're adding all the molecules to the media. A complex media, which has growth and growth factors and trace elements added by something like a beef extract, which we don't know what's all in it. A reducing media, something we use for growing obligate anaerobes. A selective media, this is one where we select for the organism of interest and it selects against other organisms. A differential media, which allows us to see differences in different cells. And then lastly, an enrichment media, which is selective, but it's designed to increase the numbers of a desired microorganism to detectable levels. Any question about any of that? I'm not going to talk about that. All right, so let's talk a little bit more about streak plating, i.e. streaking for colony isolation. What you do is you do this to obtain a pure culture, or you could streak out a pure culture. And what you do is you streak out in quadrant 1, and that will be highest in concentration up here and slightly lower down here, but that dilution will not be that great from this end to that end. Any question about that? You then take the loop and sterilize it by putting it in a Vecto incinerator, and then you need to streak on quadrant 2. Why is this backwards? Let's say you start here, and in reality you shouldn't streak here because there's nothing on the loop. You want to streak across quadrant 1 3 or 4 times to pick up cells from quadrant 1 and then deposit them in quadrant 2. So there were no cells on the loop, so quadrant 2 will be greatly reduced from quadrant 1. And as you can see in quadrant 2, we're beginning to get isolated colonies. And then you sterilize the loop to kill off the cells from quadrant 2, and then streak it out quadrant 3. And this is only showing you you're going through quadrant 2 one time. You should go through quadrant 2 the bottom part of quadrant 2 3 or 4 times. Where quadrant 2 is low in numbers. So this is going up fairly high in numbers. We should only be doing quadrant 2 near the bottom portion of it. And streak it out into quadrant 2 3 or 4 times. And then quadrant 3 will be greatly reduced in cell numbers from quadrant 2, and then streak it out. And someplace in the streaking, you will get isolated colonies. In this case, it's in quadrant 3 or sector 3 where we're getting isolated colonies, but it doesn't matter where we get the isolated colonies. They could be in quadrant sector 3, in sector 2, or sector 1. Colony streaking only works well when the organism of interest has at fairly large numbers compared to the total number of cells in the quadrant. And as you can see in this streak here, let me blow that up, we have 2 cells in this, a reddish one and a yellowish one. And as you note, we could have isolated a pure culture of the red one, but this yellow could be contaminated, that yellow could be contaminated. We don't have the yellow really isolated. That colony might be isolated. And what you would do is you'd take an isolated colony from here, this one or that one, and then streak it out for colony isolation a second time. And then when we get an isolated colony from 2 colony isolation events, we then say there's a really good chance that that colony is a pure culture, over 95% pure. This culture looks like it's pure, but it's not 95% pure, meaning there could be a few yellow cells in there. You'd want to streak it out again. If this colony was formed by more than 1 cell, like by a clump of cells, and you only need 1 yellow cell in there, and then that's a mixed culture. And so, like I said, you'd take this out and streak it out for colony isolation a second time, and then that isolated colony over here would be likely to be a pure culture. And we take from that colony over here to set up our stock culture. Any question about any of that? All right, let's move on. There are 3 ways that bacteria can grow. Most bacteria grow by binary fission. A few species of bacteria can bud. A yeast bud. A bud is where a cell puts off a small bud, and that small bud grows bigger and bigger. Eventually, the DNA inside the parent cell divides, and 1 copy is put into the bud. And then the bud doesn't separate from the parent cell until that bud is about the size of the parent cell. And then it becomes its own parent cell and then can start budding. So there's a few bacteria that reproduce by budding. There are also a few bacteria species that can reproduce by reproductive spores. Spores are usually at the tip of a filament, and the spores are usually smaller than the parent cell, and there will be 4 or more spores at the tip of a parent cell, and these spores, reproductive spores, can break off and go elsewhere and form a colony in another location. So there are a few species that reproduce by reproductive spores. Do not get reproductive spores confused with an endospore. A reproductive spore is a reproductive strategy, one cell makes several reproductive spores. So that's reproduction. Both the parent cell and the reproductive spores are made, and so one cell can then reproduce into 4 reproductive spores and staying the parent cell. So one cell will then split into 5. An endospore, one cell forms one endospore, and the parent cell dies when the endospore is released. That's a survival strategy. Any question about that? Let's talk about the difference between binary fission and budding. A binary fission, the cells literally split into 2. The DNA replicates. One DNA moves to one side of the cell, the other side DNA moves to the other side of the cell, and then the cell starts forming a plasma membrane and a cell wall in between them. And then the cell will divide by binary fission into 2 cells. One cell divides into 2. And here's an actual picture of a cell going through binary fission. This is yeast budding. Let's watch a little video of yeast budding. That might take a minute. Budding, binary fission, and reproductive spores are the 3 ways that cells, bacteria reproduce. Well, let's go. Oh, there it is. I have to push a button. So this cell is beginning to form a bud right here. And there's the bud. It looks like the DNA is replicating now. And it looks like the DNA is moving into the bud now. And that's still a bud on the parent cell. But around now, that bud is now completely separate from the parent cell. And this parent cell is making another bud here, and this cell is now beginning to make a bud there. Any questions about budding?
No.
All right. When we're talking about bacterial cultures, each culture has a generation time. A generation time is the time required for a cell to divide. So one cell to divide into 2, and it's the same time as the population number of the species to double. The generation time varies considerably between species. It can be as fast as 20 minutes, like E. coli can double in 20 minutes. Most bacteria have a generation time between 1 and 3 hours, but Mycobacterium leprae has a generation time of 10 days. It is one of the slowest known, and that's because the Mycobacterium might make mycolic acid, a lipid, and put it in their cell wall, and that mycolic acid takes a long time to make. Now, when we say the generation time is 20 minutes, we mean 20 minutes per generation, and that per generation is often implied. And when we're talking about the generation time, remember that it is time 20 minutes per generation. It is not something like miles per hour, meaning a unit per time. It's time per generation. So E. coli has a generation time as fast as 20 minutes per generation. And students frequently get tripped up by generation time. I'm not sure why, because we talk about the human generation time. So remember, it's time per generation. When we're looking at the generation time and bacteria growing, microbiologists could plot it on a numerical scale on the y-axis and a numerical scale on the x-axis, time on the x-axis, number of cells on the y-axis. But then the graph will look like this, and then one cell splits into two cells split into four cells into 8 cells, 8 cells into 16 cells. And as you can see, all of the points, I'm trying to show you that, I'm not getting it very well. There we go. Are all in this region here. And you can't really easily plot that. And then the graph is very complex. You'd have to have something like y equals mx squared plus mx plus b, is that it? Something like that. A very complex equation. So biologists like to use a semi-log plot where we plot the log number of cells on the y-axis and then the time as a numerical scale on the x-axis. And then the graph looks like this. We can split out these plots in this region now. Divide them out because they're not all lumped together right in here. And then the next one will be like right here, whereas this one, the next point, it'll be on the ceiling, okay? So we can get more points on a graph if it's semi-log. And we split these out so they show up rather than being all clumped together here. And then this equation is very simple. It's y equals mx plus b, a linear equation. So that's why microbiologists like the semi-log plot of the generation time. Any question about that? All right, we're going to end here because I'm now over and I have not finished this lesson, so we're a little bit behind schedule. All right, any questions? If not, I'll see you in the lab if you have questions for the lab at 6:30. I'm only going to be there for 15 minutes. Unless there's lots of questions. All right? All right, I'll see you.